Dear students, look at the screen. These are Drosophila Melanogaster. Here two diagrams are observed. Okay, the smaller one, this one is male Drosophila, and the bigger one is the female Drosophila. Okay, you know, Drosophila are extensively used in experimental genetics. Why? Because it has many, or they have many advantages. The first advantage is the flies are very tiny. These are not harmful, and we may culture them easily on synthetic medium in the lab. Okay, that is, we can easily culture them in the laboratory. This is the first advantage. The second advantage is regarding their lifespan. They have a very short lifespan, and due to that reason, within few months or few days, we can observe a large number of generations. Large number of successive generations can be studied easily within few months, because the average lifespan of male is 33 days, and in case of female, that is 26 days. Next point is that their reproduction rate is very high. That is, in a single mating, a large number of progeny are born. It means that you can get, or we may get, a large number of progenies by a single mating. Okay. Then the most important point is that in case of their somatic cell, the twice n is equals to 8, that is n equal to 4. Their ploidy is very less, only 4. It means that a body cell contains 8 chromosomes, and due to these less number of chromosomes, it is very easy to study the phenotypic and genotypic characters. The other two advantages are heredity variations many, and they can be easily studied under low power microscope, and moreover the male and female can be easily distinguished. The smaller fruit flies are the males, and the bigger one are the females. Okay, now have a look at the chromosomes. Now the chromosomes are in our screen. Okay, these are from the male. These 3 pairs are autosomes, and this pair is sex chromosome. It is same in the female also. These 3 pairs are autosome, and the lower pair is sex chromosome. Okay, now if we numbering them, then it will be like this. this pair is the first one, then it is the second pair, then third pair, and these two are the fourth pair. Okay, again, first pair, second pair, third pair, fourth pair. In case of female also same, pair 1, pair 2, pair 3 and pair 4. Okay, you see, the autosomes in case of male and female are same. The first pair of male and female are morphologically similar. It is same to the second pair also, and third autosomal pair also it is same. Okay, that is, in case of the autosomes, these are morphologically same in male and female. And interestingly, if we study the sex chromosomes, then these three, one from the male and two from the female, are morphologically similar, that is homomorphic. Okay, but this one is different. That is, in case of the female, the two sex chromosomes are morphologically similar, but in case of male, these two pairs are not same morphologically. They are quite different. One is longer and another one is comparatively shorter. The longer one, this one, is termed as Y chromosome, because Wilson in the year of 1909 proposed Y chromosome for it as a name. He named it Y chromosome. Clear? So we may say that the X of the male is homomorphic to the two X chromosomes of the female. Clear? Again, the X of this male is homomorphic to the other two X chromosomes of the female Drosophila. Clear? But Y is not homomorphic to the X. It is heteromorphic. Clear? Okay. Now, if we consider the ploidy, then it is clear from the figure that here the total number of chromosome is 8, that is 2n equal to 8, so n equal to 4. It is same in case of female also. Okay, that is, in case of Drosophila the linkage group is 4. Okay, now if we study the expression of the male and female Drosophila, then it is clear that the male Drosophila is described as, this one is described as 2a plus x y, where a equal to 3. That means 2 into 3 6, 7, 8 chromosomes are present in a cell, in a body cell, somatic cell. Okay, and how the female can be expressed? The female will be expressed as 2a plus XX, where a equal to 3. Okay, now regarding the formation of gametes, what happened? Take this one, this one, this one, and from here M1. It will be like this: 3 autosomes and 1 X chromosome. Clear? Okay. That is A plus X. Another one will be A plus Y, where A equal to 3. It means that the male is heterogametic because these two male gametes are different. One is A plus X, another one is A plus Y. Clear? But in case of female, it will be like this: A plus X. If we take this one, this one, this one, and from one, so there will be 3 autosomes and 1 X chromosome. Okay, and remaining autosomes and sex chromosome, they will form the another gamete. So it will be A plus X again. That is why the female are called homogametic, whereas the male are called heterogametic. Clear? Male Drosophila is heterogametic and female Drosophila is homogametic. Okay. Now let us take a look on the X chromosome and Y chromosome again. In Drosophila, the Y chromosome is inert. It is clearly written that Y chromosome is inert because the Y chromosome is totally heterochromatic. Okay, that is, the Y chromosome is heterochromatic, and due to this heterochromatic nature, the Y chromosome is inert. Clear? So in case of male Drosophila, it is having 1 X and 1 Y chromosome. X is active whereas the Y is inert. Why it is inert? Because it is totally heterochromatic, and you know that heterochromatic chromosomes are genetically inert as a repetitive DNA sequence is found in it. That is, Y chromosome, if we study carefully the Y chromosome of Drosophila, then we will observe that there are repetitive DNA sequences in that chromosome. That is why it is heterochromatic or inert. But the X chromosome of the male as well as the X chromosome of the female, in case of female there are are two X chromosomes. So the X chromosomes are active because they are euchromatic, mostly euchromatic. It means that it is more active as it is made up of unique base sequences. If we study the X chromosome, it may be from the male Drosophila, or X chromosomes may be from the female Drosophila. These X chromosomes are euchromatic and they are made up of unique base sequences. Due to this property, X chromosome is active, whereas Y chromosome is inert, and Y chromosome's inertness is only due to its heterochromatic nature, because that one is made up of repetitive DNA sequences. Clear? Okay. Now let us look the expressions of X and Y, or expressions of the genes present in X and Y. Okay, let us study. Regarding the gene expression, in males, all genes of the single X chromosome are expressed. We know that in case of male, X and Y chromosomes are present. All the genes of the X will be expressed. Okay, and there is no any allele in the Y chromosome. So the genes present in the X may be dominant or may be recessive. Either dominant or recessive, all will be expressed, because there is no any other allelic genes in the Y chromosome. Okay, but in case of female, it is having X and X. If any gene is here, its allele may be there, and that is why the expression in case of female may be homozygous or it may be heterozygous. But in case of male, it was hemizygous, because out of the two sex chromosomes, the expressions, genetic expressions are expressed only by the X, not by the Y. That is why it is called hemizygous. So the male Drosophila is hemizygous and the female Drosophila is homozygous or heterozygous. If the two alleles are same, just like capital T of a pea plant, like that, if they are dominant in the both cases, then it will be homozygous, and if one is dominant, another one is recessive and present in different X chromosomes of the female Drosophila, then it will be termed as heterozygous. Clear? Okay. Now we will talk about the determination of sex in Drosophila melanogaster. Okay. Calvin Blackman Bridges crossed triploid female Drosophila to normal male Drosophila. Okay. What is the genotype of the triploid female Drosophila? It is thrice A plus XXX, or we may say 3A plus 3X. And what is the genotype of normal male? It is 2A plus XY. Clear? So he made a cross between them. Now look at the functional gametes. In case of the female triploid Drosophila, this one, the gametes may be 2AXX, then other will be A plus X. Clear? So 2A plus A, it is thrice A. 2x plus x equal to 3x. Okay, like that. So again, the first functional gamete may be 2a plus xx, second one is a plus x. If the third one is 2a plus x, then the fourth one will be a plus xx. Okay, these are the functional gametes obtained from the triploid female Drosophila, and the functional male gametes from the normal male Drosophila are A plus X and A plus Y. Clear? So put these functional gametes in the checkerboard, and then you will get the new phenotype as well as the new genotype of the offsprings. Okay, so what will be the total number of offspring? Total number of offspring will be 4 into 2 equal to 8 in a single cross. Okay, let us see. And regarding this, we have to study about the, or we have to look after about the sex index. What is sex index? It is the determinant of the sexes in Drosophila, and it is calculated by this formula. Number of X chromosome in the individual is divided by number of autosomal sets in the individual. Okay, or simply we may say that the ratio of X and A is termed as sex index, and this is also termed as genic balance. Clear? So by the crossing we will get few genotypes, and after the getting the genotypes we will study the sex index, and with the sex index we will designate the different sexes of these 8 individuals. Okay, let us see. So this is the cross. This is triploid female and its genotype is 3a plus XXX, and diploid male, here the genotype is 2a plus XY. So the gametes, male gametes will be A plus X and A plus Y, whereas the female gametes, the functional female gametes will be of 4 types, and these are like this. What is that? The first one is (2a plus XX) and second one is A plus X. Okay, then the third female gamete is 2a plus X, and the fourth one is A plus XX. Again I am showing you. These are the male gametes, A plus X and A plus Y, and these are the female gametes, 4 female gametes. First one is 2a plus XX, second one is A plus X, third one is 2a plus X, fourth one is A plus XX. Consider this one and this one. So 2A plus A, it will be 3A. Then sex chromosome from here 2X, and here again it is X, that is it will be thrice X. Okay, that is 3A plus XXX. Similarly here it will be 3A plus XXY. Now let us calculate the index. The index is determined by X by A. So what is the total number of X here? It is 3. And what is the total number of autosomal sets? Here it is again 3. So 3 by 3 equal to 1. Here the index is 1. Clear? Similarly, in this block, the index will be 2 divided by 3. It will be 0.67. Clear? Consider this one, this block and that block. Here the index will be 2 by 2, that is 1. Okay, and here it will be, in this block it will be 1 by 2, that is 0.5. Similarly, have a look in other blocks also. In this block the index will be 2 divided by 3, that is 0.67. What is about this block? It will be 1 divided by 3. Okay, and the result will be 0.33. And consider this one. Here the combination genotype is 2A plus XXX. So the index will be 3 divided by 2, that is 1.5. And in case of last one, let us see. In this last block it will be 2 divided by 2. Index is 1. So these indexes are very important. Okay, because with the help of these index we can determine the sex of the Drosophila. Let us see the index. If the result or index is less than 0.5, it will be super male or meta male. We have seen that in the block there was 0.33, one case. Okay, and that will be super male. And if the index is equal to 0.5, then it will be normal male. And if it is in between 0.5 and 1, then intersex. Again, if it is 1, then it will be normal female. And if it is greater than 1, then it will be super female. 0.5 and 1.0. Okay, it is like this. Here it will be super male, then normal male, then intersex. At 1 it will be normal female, and if it is more than 1, then it will be meta female or super female. Clear? Okay. So these are the results. From the first block, we got the index as 1. So it will be triploid female. And in the second block, it is, the index is 0.67, hence it is triploid intersex. Have a look at the cross, then it will be clear. Here the index is 1, so it is triploid female. Then here the index is 0.67, so it will be intersex. In the third block, the index is 1, that is normal female. In the fourth block, the index is 0.5, normal male. In this block, the index is 0.67, again intersex. In this block, the index is 0.33, super male or meta male. Here the index is 1.5, meta female or super female, and at last the index is 1, that is normal female. Clear? Okay. Now have a look on the X, Y and A regarding the maleness and femaleness of Drosophila. From the above cross, Bridges postulated that X chromosome determines femaleness and autosomes determine maleness. Again, femaleness is determined by X chromosome and maleness is determined by autosomes. Okay, clear? Study these cases. Case number 1. The genotype is 2A plus XX. So what will be the index? Index will be 2 by 2 equal to 1. That is normal female. If we add X chromosome by suitable crosses, then the result will be 2A plus 3X. So what will be the index in this case? The index will be 3 by 2, that is 1.5. Okay, that is meta female. Clear? So from this case it is clear that if we add X chromosome to this genotype, then normal female will be converted into meta female. Okay, that is femaleness is increased. Have a look on the second case. Consider this case. Here the genotype is 2A plus XY. 2A plus XY. So what will be the index? Index will be, for X it will be 1 divided by this 2 equal to 0.5. That is normal male. Okay, if by suitable crosses, if we add autosome, a set of autosome, if it is increased, then the result will be 3a plus xy. So what will be the index? Index will be 1 divided by 3 equal to 0.33. That is super male. Clear? That is, from this case it is clear that the maleness is increased by adding a. Okay. Let us consider another case. Previously, we have seen that by increasing X and A, the femaleness and maleness are increased respectively. Okay. Now, let us study the another case. The case number 3. Here the genotype is 3a plus xx. So the index is 2 divided by 3, 0.67, and 0.67 is intersex. Okay, and if we increase x, it will be 3a plus 3x, and the index will be 3 by 3 equal to 1. That is triploid female. That is, an intersex Drosophila can be converted into female Drosophila by adding X. Okay. Whereas, if we study the case number 4, then here the genotype is thrice a plus 2x, that is the index is 2 by 3 equal to same 0.67, that is intersex. Let us increase a, then it will be 4a plus 2x, and what will be the index? Index will be 2 by 4 equal to 0.5. That is, an intersex Drosophila can be converted into normal male. Again I am showing you. This one is intersex Drosophila and that one is also intersex Drosophila. If we add X to this one, it will be converted, the intersex Drosophila will be converted into triploid female. That is, femaleness is increased by adding X, whereas intersex has been converted into male by adding A. That is, if we increase or if we add A to the intersex Drosophila, then that intersex Drosophila will be converted into normal male. Okay, so from these 4 cases, it is clear that the maleness is increased by adding A and femaleness is increased by adding X. That is, the maleness is solely dependent upon the autosomes and femaleness in Drosophila is solely dependent upon the X chromosome. Clear? And the total index, that is all the indexes shown the different types of phenotypes. Okay, and these phenotypes are totally dependent upon the ratio of X and A. That is, X-A ratio is called genic balance, and we may say that the sex in Drosophila Melanogaster is dependent upon genic balance. If X is added, then femaleness is increased, and if A is added, then maleness is increased. At last, again I am repeating, the maleness of Drosophila is dependent upon A, and femaleness of Drosophila is dependent upon X chromosome. Okay, thank you.